So without further ado, today we will be hearing from Dr. Jason Choi. He's a current assistant professor at the Center for Translational Medicine at Thomas Jefferson University. And as a postdoc, Dr. Choi observed some pretty unique findings in the sense that he observed that within germline point mutation of mouse models of the LMNA-derived cardiomyopathy, that cytoprotective autophagy in the heart was impaired. And through this finding, he was able to get four publications, three of which were first author. So that is very impressive. And currently now, as an assistant professor, his research focuses on understanding the pathogenic mechanisms underlying cardiomyopathy from mutations within the LMNA gene, which is known to encode lamin AC. At Thomas Jefferson University, he focuses on observing cell-specific phenotype within these models. And through this work in his lab, he has already published 4 publications, and I'm really excited to hear more about your research. So with that, I will go ahead and pass it off to you, Dr. Choi. Go ahead and share your screen.
Well, thank you for the kind introduction, as well as the invitation to share some of the work that we've been doing. Let's see, screen 2. There we go. So when I agreed to this date for a presentation, I didn't realize I was putting myself in an unfortunate position of going after sort of 2 top scientists. So it's going to be a hard act to follow, but I'll do my best. So what I hope to achieve is to sort of convince you guys of the story that we're building through experimental evidence using just standard techniques. There's nothing super cutting edge or fancy here, but what I hope to achieve is to, as I said, convince you of the story that we're building with regard to the pathogenic mechanisms leading to dilated cardiomyopathy arising from LMNA gene mutation. So as this crowd knows, I don't really need to go that into background with regard to lamins. And my favorite intermediate filament is nuclear lamins, which was sort of continued on from Dr. Howard Warman's lab when I was a postdoc in his laboratory. And when I first came here, I started thinking about the pathogenesis. And when you look at the heart is a complex sort of heterocellular organ, which is composed of multiple different cell types, and some of which aren't even listed here. And we wanted to focus on 2 specific cell types, one which is cardiomyocytes, which are the force-generating cells of the heart, and the cardiac fibroblasts, which are sort of the central mediators of fibrosis or injury response. And sort of the interplay between these 2, what mechanisms sort of underlie the interaction between these 2 cell types to engender disease. And as Clara already mentioned that we've discovered, and there was also several other groups that shown that autophagy is sort of involved. And actually, the first hint that autophagy was involved is actually a paper in 2019, which Dr. Giselle Bon was a co-author. And in their mice, they looked at, I believe it was the fibroblasts that show that when there's sort of a impaired autophagy, and they saw a large sort of vacuoles around the nucleus, and then they thought that nucleophagy was occurring. And then if you facilitated this process, that it actually leads to improved outcome. And they also saw that in the patient samples, similar things were happening. And then subsequently, we and others have shown in the murine models that autophagy impairment actually contributes to the disease pathogenesis, as well as subsequent other studies in other model organisms, as well as in human patients. So to sort of start studying this process, as Clara already mentioned, that we approached it in a cell type specific way. And at that time, there weren't many studies looking at cardiomyocyte specific deletion. So when I first started here, we generated a Cree driver line, which is actually a bicistronic construct that expresses both the tamoxifen regulatable Cre ERT connected by internal ribosomal entry site to a GFP fused L10A, which is a ribosomal protein, and this I'll get into it later. Just to mention that a lot of the most of the cardiomyocyte data that I'll be presenting has been published. But then the cardiac fibroblast story it's all unpublished. And for this model, we have two Cre driver lines in our laboratory. One is collagen 182 Cre ERT that was made by Dr. Christopher Denton a while ago, and periostin MCM Cre, which was generated by Dr. Jeffrey Mulcahy. And the unique element of periostin MCM is that you could target specifically activated fibroblasts because the periostin promoter isn't really active until the fibroblast gets activated. So this is just a validation that our mouse model, we've identified two founder lines. They express both GFP as well as Cre ERT. Shown on the right side of that is direct fluorescence imaging of GFP L10A. And what you notice here with when we co-stained with sarcomeric actin to ensure that the expression is limited to cardiomyocytes, you see these punctate structures in the nucleus, which is their typical localization in addition to the sort of a diffuse cytoplasmic staining, because L10A or ribosomal protein is also enriched in the nucleolus. And then the right here is just the qPCR analysis showing that the transgene mRNA expression is pretty much limited to just the heart itself. So now having this model, we wanted to see what happens when we delete LMNA C, specifically in the cardiomyocytes. And this is the deletion strategy that we used. We deleted the adult cardiomyocytes to sort of assess the acute effect of lamin knockdown in cardiomyocytes, because many of the conditional studies, or if you look at if the deletion occurred in utero, it's very likely that there's postnatal developmental defects that can impact what you see in the heart later in life. So we wanted to sort of delete lamins at 12 weeks of age by injecting tamoxifen. And then at weeks 2 and 4, after the final tamoxifen dosing, we performed echocardiography, which is cardiac ultrasound to look at cardiac function and histopathology and survival. So first, to show what happens with lamin knockdown or lamin depletion, A-type lamin depletion in cardiomyocytes, we see that at 4 weeks, only the homozygous deletion, but not the heterozygotes, show significant reduction in fractional shortening, which is a measure of a percent reduction of the ventricular dimension going from when the heart is relaxed during the diastole to systole when the heart the left ventricle contracts. So that dimension, that reduction in size of the ventricular dimension is significantly reduced, indicating that the heart cannot contract as well. We do not see that even at 4 weeks when the cardiomyocytes retain a single copy of lamins, or at 2 weeks, which is probably too early for the disease or the impact on the heart to be evident. And when we looked at, we also performed similar studies at 12 weeks of age, the collagen 182 Cre as well as periostin Cre, and we really went in thinking that we were going to see a phenotype with just the deletion of if we delete lamin AC in the cardiac fibroblasts, and to our surprise, we didn't see any phenotype in these mice, and we've kept these mice, going until 6 months later. They survived; they do not die. But we can detect the recombined allele in the heart as well as in many tail fibroblasts, even after 6 weeks after or 6 months after tamoxifen deletion, which was very surprising. So in this case, what we saw was that only the homozygous deletion of A-type lamins in the cardiomyocytes show a robust phenotype. And then when we looked at the histopathology, it kind of sort of matched what we saw we started seeing some abnormal histological, abnormal histological staining in the myocytes starting from 1 week, but by and large, it was mostly normal. At 2 weeks, we start to see small pycnotic nuclei pop up, as well as sort of myocytes that looks to be undergoing what the pathologists call cytoplasmic lysis. And by 4 weeks, we see myocyte swelling, abnormal nuclei, which is also present at 2 weeks, but the number of the abnormal nuclei increases as the mouse gets sicker. And with the same story with a fibrotic staining, collagen staining with Sirius red, and the quantitation is here, we see this progressive increase in fibrosis. This is sort of confirmed by qPCR analysis when we looked at cardiac stress hormones, NPPA and B, which encode precursors to natriuretic peptides to sort of relieve this stress, the stretch stress from the heart by acting on the kidney to get rid of fluids. CTGF and periostein, which are matricellular proteins, which are highly expressed in activated fibroblasts, start to go up at 2 weeks and 4 weeks. And the same story with a lot of the collagen isoforms, as well as the fibronectin, that we start to see increased expression starting from 2 weeks, and by 4 weeks, the expression of these proteins is dramatically increased. And when we looked at survival, we saw that they survive with a median survival of 33 days. And ever since this paper came out, there were a couple of other papers where they looked at A-type laminin deletion in adult cardiomyocytes. And the data is extremely consistent in that typically around a month after induction of laminin deletion, you get severe cardiomyopathy accompanied by robust fibrosis. Now, when we looked at the histological staining for the cardiac fibroblast-specific deletion, these mice don't die. As I mentioned, they live like a normal mouse, and their histology is virtually normal. So when we took a closer look at the adult cardiomyocytes, the myocytes-only deletion, what we started seeing was this nuclei that's sort of different than what we were used to. And at least in my memory at that time, there were two clear studies where the first one was from 2004 using this sort of what they thought was initially a knockout mice, but that ended up expressing a small truncated form. But still, it phenocopies a hypomorphic or knockout mouse model. That was generated by Dr. Colin Stewart. And the H22P point mutant mouse model, which was generated by Dr. Giselle Bond, both of which showed this sort of long, elongated nuclei. But that's not what we were seeing. We were seeing these really abnormally shaped, weird-looking nuclei, which we weren't really sure what to make of, or maybe whether it actually even matters. This is actually kind of relevant if you sort of pay attention to the normal morphology of an adult cardiomyocytes that kind of look like this, where adult cardiomyocytes are typically bi or trinucleated, and around the perinuclear space, it sort of has this fusiform shaped area where it's packed with perinuclear mitochondria as well as the Golgi. And I'm pretty sure that the perinuclear ER is there, but it's just very difficult to visualize with EM. And shown on the bottom of this EM image is the mitotracker of an adult cardiomyocyte, mitotracker staining of adult cardiomyocytes, where you can clearly see this sort of enhanced or brightened area perinuclear shape. It's a nice long fusiform shape. And the idea, there's some data in the literature indicating that these perinuclear mitochondria actually plays an important role specifically for a nuclear function. But when we looked at a little closer to the EM at the perinuclear space, what we notice is that in the control mice, we get this beautiful sort of nice round nuclear envelope towards the polar ends, which is an area of high curvature, which is prone to nuclear damage, which was shown by Dr. Dennis Disher. And then you see perinuclear mitochondria as well as these Golgi apparatus nearby. But when we look at the 2-week post-TAM, this is sort of typical of what we see. We get this really weird damaged nuclei with these 10 nanometer or 100 nanometer vesicles that we're not really sure exactly what they are, but only thing that we could say is that there's this severe damage, and we have data that I won't be showing here, that the perinuclear mitochondria sort of dissipates or goes away. It possibly through mitophagy, but we know that once we've taken adult cardiomyocytes and stained them with mitotracker after A-type laminin deletion, then we see that there's far less perinuclear mitochondria there. But this story will be focusing on the Golgi, which was our main story for the story that we published last year, is that we get this sort of Golgi apparatus that's right around the nuclear membrane. And by 2 weeks post-TAM, tamoxifen, we get this fragmentation and start to see dilation of the Golgi stacks, which gets significantly more obvious by 4 weeks when the heart is already severely damaged. So why does this matter? Most people think of Golgi as sort of a trafficking hub for intracellular transport, vesicular transport, but Golgi also serves various other functions, importantly, the activation of stress responses that depend on proteases that exist in the Golgi, such as Site1 and Site2 proteases. So if there's when there's a stress response, such as Golgi stress, or even ER stress, various different stress intermediaries they hang around in the ER, they migrate, they translocate to the Golgi, and then they get cleaved by Site1 and Site2 proteases that activates, that releases a transcriptionally active form that goes into the nucleus to transactivate gene expression to alleviate stress. In addition, there's a protein GNP-TAB, which actually is it's a glycosylating enzyme that tags mannose-6-phosphate to various proteins that are destined to the lysosomes. And this protein needs to be processed by Site1 proteases to function. So therefore, Golgi, the intact Golgi is actually required for a proper lysosome biogenesis. And as Clara has mentioned before, that we've studied autophagy impairment, and we reasoned that, oh, maybe this is the reason why we're seeing this. And we looked at the hearts of our mice to look for evidence of impaired autophagy, and we see that typically at 4 weeks, we start to see it sort of accumulate from 1 to 2 weeks, but it's very obvious by 4 weeks where we see accumulation of lipidated LC3B as well as P62, which are components of the autophagosomes. And then the fact that those two are sort of hanging around indicates that autophagy are not being degraded properly by lysosomes. And to further drive that point, when we directly impair the Golgi, and this is these are C2C12 cells that we use as a in vitro model. C2C12 are muscle myoblast cell lines. And then if you knock down these two proteins, GORASP1 and GORASP2, depletion of these two proteins can basically damage the Golgi. And when you deplete both of these through different combinations of short hairpins for GORASP1 and 2, we could see that the P62 as well as LC3B lipidated form starts to accumulate. And when we overexpress EGFP, LC3B to kind of see how they're localized in the cell, and we stain also with P62, you could see that LC3B co-localizes with P62, indicating that these are true bona fide autophagosomes. So you impair the Golgi, you impact autophagy. Now, would you see something similar if you were to damage the nuclear envelope. And to do this, we've taken mouse embryonic fibroblasts that were isolated from LMNA flox mice, and then we hit them with adenoviral Cre to delete A-type lamins in vitro. And then we stained them for RCAS1, which is a protein that's localized to the Golgi that we use as a surrogate for Golgi area or Golgi size. And in MEFs that were infected with an adenovirus carrying the adenovirus carrying blank construct, we see typical nuclei that's nice and round, and with the RCAS or Golgi staining that are typically like sort of a crescent shaped at the one side of the nucleus. But with lamins knockdown, we see these abnormal, weirdly shaped nuclei form, which has been shown many times in the past. And then what we found is that RCAS1 signal basically goes away. And we could see that better. And when we actually used these images, the data from here, and we used the ImageJ plugin, which measures, it's an automated nuclear irregularity index plugin that basically measures the area and then basically measures the shape of the nucleus and assigns basically how messed up, how irregular the nucleus is. And then the higher the nuclear irregularity index, the more messed up nuclei, as you can see here. And after adenoviral Cre, we see that this increase in nuclear irregularity index shoot up. We've done this with, it's not from the effect of Cre. We've done this many times with just the non-floxed wild-type MEFs that were also infected with Cre, and they do not impact the nuclear shape at all. So it's not the effect of the virus, viral infection. And when we actually take sort of the RCAS area and plot it with nuclear irregularity index, sort of a negative correlation forms where low RCAS area is correlated with highest nuclear irregularity index, which suggests that the more messed up the nucleus, there's less RCAS1 or Golgi footprint there, suggesting that there is the abnormal nucleus sort of underlies Golgi damage. And this is sort of a preliminary data that we're starting to look at. We looked at S1P levels by Western blot in MEFs that were treated with adenoviral blank or adeno-Cre, and that were also treated with increasing dose of this drug called monensin, which is actually a Golgi stressor. And what we see is that with just the lamin depletion, lamin-AC depletion, we get reduction of S1P. Cathepsin D is one of the enzymes that are targeted to the lysosomes, and lamin depletion leads to a reduction in this protein that's further reduced by increasing dose of monensin as well. RAB5 and 7 stain for early and late endosomes, and these don't seem to be impacted that much by lamin deletion, but they do seem to respond to Golgi stress monensin, increasing dose of monensin. And shown on the right, you see the cells these are cell MEFs that's been treated with adenoviral Cre. And what we typically see in this snapshot in time, which is, I believe it was 24 hours after 48 hours after adenoviral Cre, that you start to see some of the cells clearly losing A-type lamins, and a lot of these cells don't really have much of any vesicles that are being stained with lysotracker. The yellow arrow shows this large sort of vacuole that the original 2009 paper has seen, indicating that it might be nucleophagy that's occurring. But those cells that retain A-type lamins and that are still mostly normal looking, still has a lot of vesicles that are acidified. So kind of switching gears, as I mentioned, our mice is bicestronic. It also expresses GFP-L10A, which, as I said, is a fusion protein of GFP with L10A, which is a ribosomal protein. And when the construct or this protein is expressed, it actually takes normal part in ribosome formation, and it actually undergoes translation. And this GFP tag allows us to pull down using GFP antisera and to enrich for translating RNA, specifically in cardiomyocytes. So that's what we did here. We injected, again, we deleted lamins, and at 2 weeks, we harvested the hearts, lysed them, and then did TRAP, or translating ribosome affinity purification. And then we sent it out for RNA-seq. And before we did that, we did some control experiments just using qPCR, looking at various different genes that are cardiomyocyte specific, cardiac fibroblasts or endothelial cells. And then the more redder the genes are, that means that it's much more enriched. And that's what you see here, is that a lot of the cardiomyocyte specific genes are much more enriched than the fibroblasts or fibroblast specific genes or endothelial cell gene. So it shows that our TRAP system works in enriching cardiomyocyte specific translating RNA. And when we submitted for sequencing, our earlier preliminary data show that the two proteins were specifically enriched, mediator MED6 and MED25. Now, we confirmed that their protein expression in the hearts of these mice, both in founders line 1 and 2. And what mediator is what many people consider it to be the endpoint of signal transduction, where transcription factors that are induced by external or even intracellular stimuli will go into the nucleus, and when they bind to the enhancer regions of the promoter, it doesn't directly interact with the basal polymerase transcriptional machinery. They actually relay their signal to this complex of evolutionary conserved family called mediator complex, and it's this about over 30 complex superfamily of mediators that basically relay the signal from the transcription factors to the basal polymerase machinery. Now, mediator 6 seems to play a bigger role in the general basal transcriptional activation, whereas mediator 25 seems to be more selective. It's involved in lipid metabolism, as well as a paper showing that both mediator 25 and 6 can both bind to ATF6, which is, if some of you may know, is a member of one of the canonical branches of the ER stress pathway. So we thought that maybe, there must be ER stress going on, which we really didn't see. We only saw a selective activation of PARC ATF4 pathway, but not the more cytoprotective pathway that are mediated by ATF6 or even XBP1, which we later found out is that selective activation of ATF4 CHOP pathway is actually emblematic of what you see during Golgi stress. So following that same theme, we generated knockdowns of C2C12 cells depleted of MED25 using 2 different short hairpins. And we have the same data with a different cell type, 3T3L1 fibroblast. And we basically see the same result, but I'm just showing the C2C12 here. What we did was that when we depleted MED25 and we hit them with increasing dose of, again, Golgi stressor monensin, and what we found was that the cells that have MED25 knocked down are more susceptible to the toxic effects of monensin, whereas the blank control cells that has been infected with a lentivirus carrying a blank construct were more resistant. And we thought, okay, it seems to suggest that MED25 confers protection to Golgi stress. Is this possibly working through site 1 proteases? And we tested this by treating the cells with this compound PF429242, which is a site 1 protease inhibitor. And by itself, the cells don't do much. We didn't see a lot of cell death. But when we co-treat now with monensin, which I believe here was the 20 micromolar concentration, with the site 1 inhibition, now we start to see even the blank cells are highly susceptible to cell death. More interestingly, the way these cells were dying was very striking, in that when we hit them, when MET25 knockdown cells were hit with high dose of monensin, these cells die by these accumulating these vacuoles in the cytoplasm, which we did not see that much with the blank control cells. When we hit these cells with the Site1 inhibitor, the cells weren't happy, clearly, and they swell up, but they were still sort of alive and viable. However, when we, as I showed here, if we hit them both with monensin and Site1 inhibition, even the blank control cells now die by accumulating these large vacuoles in the cytoplasm. And this was striking to us because if we look at the mouse hearts, at 4 weeks, at cardiomyocyte-only deletion, these hearts are filled with huge, like significant vacuolation. And the implication is that there are similar processes happening in the cells, in the C2C12 or in vitro culture model, as well as in the in vivo, in the hearts in vivo. Well, I better hurry up. So the conclusion thus far is that LMNA depletion in adult cardiomyocytes lead to rapid cardiomyopathy development. This myopathy is accompanied by robust fibrosis, which was the first hint that maybe the LMNA plays an important function for cardiac fibroblast function. LMNA deletion in cardiac fibroblasts in and of itself does not drive pathogenesis, which means that the cardiac fibroblast activation and fibrosis development occurs as a reaction to cardiomyocyte damage. And lastly, adult cardiomyocyte nuclear damage precedes cardiomyopathy development. So at this point, the basic sort of model is that you have lamin mutation in the myocytes leads to their damage and activation of cardiac fibroblasts that leads to fibrosis. And with fibrosis comes tissue stiffening, which imparts sort of physical stress to the myocytes themselves, which may further exacerbate the nuclear damage, which sort of keeps it in a death loop until the myocytes die. But so a lot of the data, the in vitro model that we've shown are done in either C2C12 cells or mouse embryonic fibroblasts, which morphologically, in a lot of ways, or at least for MES, even functionally, is more similar to cardiac fibroblasts. So we reason that if these processes that we observed in cardiomyocytes, and then the in vitro models that we use to sort of validate that are sort of similar to cardiac fibroblasts, maybe the similar molecular pathogenesis is occurring in cardiac fibroblasts as well. And we wanted to study that. And for anybody who's sort of studying cardiac fibroblasts, know that there's a huge technical challenges working with primary cardiac fibroblasts in vitro. And that's that just culturing them in hard plastic leads to their spontaneous activation and also spontaneous differentiation into myofibroblasts, which was shown by my title slide. After 4 days of TGF-beta treatment, this is kind of what you see from the previous inactivated or naive cardiac fibroblast. And the pathway that drives this process is actually pretty well mapped out. And it was mentioned by Dr. Wickstrom a couple of weeks ago. It's the MRTF pathway where matrix stiffness or even TGF-beta acts on ROCK that leads to the globular actin becoming filamentous, which then that strips away the globular actin off of MRTF. MRTF is now able to gain entry into the nucleus and transactivate these genes that are important for fibroblast activation. We just took chemical inhibitors to ROCK, and TGF-beta, SD208, is a TGF-beta receptor inhibitor, and Y27632 is a ROCK inhibitor. And we also added basic fibroblast growth factor to provide a strong sort of mitogenic signal, which cell proliferation is sort of always on the other end of differentiation to kind of keep them going in this proliferative state. And this three sort of supplement is actually highly effective in suppressing spontaneous differentiation, where without the supplements, you culture these cardiac fibroblasts for several days, and this is what you see. You get not smooth muscle actin, just regular actin fibers, stress fibers forming, and you can only see this through phalloidin. And you could see this sort of fibers forming across the nucleus. And with the supplements, you could actually suppress that completely. And on the bottom, when we've stained them for smooth muscle actin, you get this sort of random expression, random differentiation into myofibroblasts, where with the supplements, you could significantly suppress this process. We isolated messenger RNA and looked at, through qPCR, message expression of various genes that are involved, upregulated with cardiac fibroblast activation, such as, collagen isoforms, matriciellular proteins, and TGF-beta. And addition of the supplements significantly reduces these gene expression. And more importantly, once we've kept these cells in with the supplements for a prolonged period, and then we could actually remove them gradually, and then you could hit them with TGF-beta, and these cells still retain the capacity to differentiate into myofibroblasts. So using this method, we went ahead and did a whole bunch of studies to test the function of cardiac fibroblasts. So we have two different models for here. We have the knockdown models for cardiac fibroblasts, which we used both the human that were knocked down with lentiviral short hairpin delivery, as well as mouse cardiac fibroblasts isolated from flox mice, where we added adenocrete. And then we have the H22P line, which was generously provided by Dr. Giselle Bond, which do not need any viral manipulation. So in response to injury, cardiac fibroblasts, they proliferate, migrate, and they differentiate into myofibroblasts, and then they contract. Not always in this order, but by and large, this is the main process that we've studied. And for proliferation, we just did simple, trypan blue exclusion. And this one's in human cardiac fibroblast. But as I mentioned, we see the same thing with the mouse mouse cardiac fibroblast. After short hairpin delivery, lentiviral infection, we see that all the cells sort of slow down cell proliferation, which is typical what you see when all these cells are sort of activating the antiviral response. But then the control cells sort of recover very quickly, whereas the knockdown cells are slower to recover. And I only show day 12 here. At day 6, there's a clear knockdown. But when you get to day 15, the knockdown cells actually catch up to the control. And when we looked at the lamin expression, the A-type lamin expression, they're basically, it comes back. So what that showed us is that the cells that still, let's say if about 80% of the cells have knockdown and 20 still retain some amount of lamin AC, and over time, these cells that still retain A-type lamins overtake the ones, they outcompete, they outgrow the cells that have the significant lamin AC depletion. Whereas the H22P, from the very beginning, we saw that they cycle much slower. Then we looked at migration, just using a simple scratch wound closure assay, where you have a monolayer and you create a wound, and then you measure how long the cells take to fill that gap. And with the lamin knockdown cells, they are slower than the controls, with cardiac fibroblasts that has wild-type lamins or A-type lamins. But surprisingly, for the H22P, the trend seems to be reversed, where these cells are able to migrate faster. I'm not exactly sure what that means, but here in this case, we were seeing faster migration with the H222P point mutant. For the differentiation analysis, we just hit them with TGF-beta and looked at smooth muscle actin expression. And for the knockdown system, we didn't see that much of a difference. We saw that they differentiated, they expressed smooth muscle actin and similar kinetics. H22P model seems to show a slight reduction in smooth muscle actin, as well as some of the genes that are induced by TGF-beta, but they were sort of highly variable. Some of them seemed okay, like about comparable to wild type, other ones seemed a little lower. But by and large, they were mostly intact. And then for contraction, we did collagen pad assays where cardiac fibroblasts are sort of grown inside the collagen pad, they're allowed to polymerize, and then they're transferred onto a media containing FBS TGF beta. And then you measure the contraction. And not surprisingly, the ones with the knockdown or H22P are not as able to contract as well as cells retaining the wild type lamin expression. Now, how about in vivo when we delete both lamins in both the myocytes and in fibroblasts, effectively generating a triple transgenic, both the collagen 1A2 Cre and periostin, using this treatment strategy, it actually delays the deterioration of cardiac function. And I say delay because eventually, when you wait out until like 6 weeks after tamoxifen treatment, these mice eventually succumb to their disease. And that's shown here in the survival data, where with the collagen 1A2 cardiomyocyte fibroblast deletion as well as periostin, you can increase the median survival relative to cardiomyocyte only deletion. And the histopathology basically looks like this, where we noticed a significant reduction in fibrosis in both with both the collagen 1A2 Cre and as well as periostin knockdown. This is just showing the same trend that we see. Since I'm kind of running out of time, I'm going to go quick. The message level for a lot of these cardiac stress hormones as well as ECM protein genes are reduced in the double knockdown or myocyte fibroblast co-deletion. And it appears that the fibroblast abundance is also lower in the heart sections that we measure cardiac fibroblasts by co-staining with vimentin and PDGF receptor alpha. Now, how about the Golgi in the fibroblasts in the cardiac fibroblasts when we delete lamins? This is the lamin knockdown model where lamin positive cells were treated with adenocre. They look totally fine. Their Golgi looks good, and then when we plotted RCAS area with nuclear irregularity index, more messed up nuclei we didn't really see any really messed up nuclei. But when we deplete lamins, A-type lamins, using floxed cardiac fibroblasts with adenocre, then this sort of negative correlation as determined by Pearson R shows up. Again, further indicating that you have smaller RCAS footprint with higher irregular or more irregular nucleus. Now, more interesting thing was that when we looked at the same thing in the H22P model, we didn't really see a difference in the basal condition. You see in the wild type cells that you get this nice sort of RCAS1 signal. H22P by itself, we could see that they're altered, as you could see here. So a lot of the nucleus looks a little weird. The RCAS footprint looks a little weird, but when we did the measurements, they were pretty similar. However, after 48 hours of TGF beta treatment, we noticed that negative correlation show up. And we noticed a couple of interesting things. One is that the Golgi footprint dramatically increases, which is not surprising because the cells are getting ready to start pumping out ECM. But also the nucleus gets enlarged, and that it's possible that for the H22P lamins, that you need this sort of enlargement of the nucleus, that force to sort of make this the pathogenesis more evident. Lastly, this is the last data figure. So in collaboration with a group at Penn, Dr. Margulies and his postdoc Nez, they have this this tunable matrix called magnetorheological elastomer, where it's the matrix is infused with iron beads, and based on how much magnetic field they apply, they could make it either 10 kilopascal or 50 kilopascal. And we wanted to do this as a way to show that the physiological matrix stiffness can underlie these nuclear damage we see in cardiac fibroblasts, because all the damage that you've seen were sort of cells growing on hard plastic, which are in gigapascal range. And to do this, we sort of developed the strategy, given that these cells are primary cells, that these cells still retain contact inhibition. So when these cells were about 70% confluent, we added adenocrete, so that when the cells are starting to lose A-type lamins, that they're pretty much constricted by contact inhibition because they're fully confluent. And various studies have shown that typically it's the forces generated during migration or even cell division that produces forces that can damage the nucleus. And this strategy will limit that. So we culture for 6-7 days, and then we played onto MRE. We imaged them at 10 kilopascal, and then they were either raised to 50 or kept at 10. And then we took the image again, and then we just looked at nuclear area versus nuclear irregularity index. And wild-type lamin at 10 kilopascal and 50 kilopascals, there's good overlap, meaning that in wild-type cells, increased stiffness at 50 kilopascal doesn't really change the nuclear irregularity index. And this is a compilation of 4 independent experiments. When we look at 10 kilopascal wild-type cells with lamin knockdown cells at 10 kilopascal, we do see some increases in more irregular nuclei. However, when we compare it with the 50 kilopascal, now we start to see significant increase. And this, I didn't show the numbers, but this is about 30% of the cells that are significantly increased in nuclear irregularity. So finally, I know I'm about 4 minutes past due. So the conclusion is that based on all this data that we have, we believe that the perinuclear damage may be the main driver of cardiomyopathy pathogenesis. And you have a lamin mutation, and that mutation in a germline model or in human patients will be present in both the cardiomyocytes and fibroblasts. And it may lead to the same molecular pathogenesis, but what effect it has on the disease pathogenesis is really based on the function of the given cell type. The myocytes function is contraction, and that's impaired. Fibroblast function is fibrosis, and if that's impaired. And it's this balance between the two that we believe is sort of the driver of disease onset and or severity. The severity of the nuclear damage likely is determined by LMNA mutation itself, as sort of evidenced by the H22P point mutant model, and the extent of the collateral damage to the perinuclear space and the Golgi, which leads us to conclude that lamin AC mediate an opposing balance between cardiomyocytes and fibroblasts in driving LMNA cardiomyopathy progression. And based on all this data, we have a working model where the myocardial damage, as shown in this axis, is driven by 2 factors. One is nuclear damage or nuclear damage index that can be empirically determined. And as I mentioned, is likely determined by the mutation, the LMNA mutation itself. And then pathological fibrosis, which sort of contributes the stiffening effect that exacerbates nuclear damage. And when we plot sort of what we known from other mouse models, we get a nonlinear relationship where the wild-type mouse have no myocardial damage, 0 fibrosis, and their nucleus is very sturdy. Lamin knockdown or knockout mice have severe myocardial damage, but they don't show fibrosis. And their nuclear damage index is probably the lowest because it's really missing lamins. H22P is somewhere in between where it still expresses normal amounts, but it looks like the cardiac fibroblasts are only damaged when they're sort of activated or induced to differentiate into myofibroblasts. And CMO, which is an artificial sort of non-clinically relevant model, which has severe fibrosis, severe myocardial damage, but the fibrosis is pretty, and it's sort of away, it sort of leads it away from this curve that we have. So I think I'll stop there. And these are the acknowledgements. Many people have sort of helped us get to this point, and I'll be happy to take any questions.